So this panel that I have the honor of moderating is called Drivers of Innovation, the Costs of the Research Enterprise at Dana-Farber. And before I introduce my esteemed colleagues on the panel, I want to sort of set a context for you, because what we're going to talk about really arises directly out of Dana-Farber's mission, this sort of twofold mission where we provide exceptional, compassionate, and equitable care to all patients with cancer. And then the second part where we try to advance the understanding, the diagnosis, the treatment, the cure, and the prevention of cancer. And it's that second part, especially around understanding cancer, that depends entirely on research. And it's the reason that for 75 years, Dana-Farber has elevated research to the same level of importance as clinical care. And we remain the only cancer center that puts research and clinical care on an equal footing, 50-50. Why are we the only cancer center that does that? Well, it's because the financials are impossible. Michael can tell you that. Let me give you some numbers. Last year, Dana-Farber spent over $500 million, $500 million on research. And every year that number increases. And that number increases not just because of inflation. That number increases because every year there are new opportunities that we need to exploit. Now, you may well think that most of the funding for those expenses comes from the federal government, from the National Institutes of Health, or the NIH. And you'd be wrong. The NIH provides only about 30% of the funding that we use to pay for research. And that proportion is decreasing every year. Let me hasten to point out that that is not a negative reflection on Dana-Farber's faculty. Quite the opposite. In fact, when we last looked, it turns out that Dana-Farber faculty bring in more NIH dollars per individual investigator than any of our comparator institutions like Memorial Sloan Kettering or MD Anderson. The problem is that research keeps getting more and more expensive, and the NIH budget just doesn't catch up. So one other way to think about this is that research is a very capital-intensive activity, just like it is in pharmaceutical companies and in biotech companies. But unlike those entities, we don't have access to the biggest funder of R&D, which are capital markets. So what do we do instead? We turn to foundations, we patent our discoveries, we try to get licensing revenue, we try to get royalties, but it still isn't enough. So how do we make up the difference? It's you guys. Seriously, it's philanthropy that makes up that difference. And that's why we always say that our donors and our friends should feel a tremendous sense of pride, and I would even say ownership over the exciting research advances that come out of our institution. And so to talk more about that, we have two incredible people sitting here. I mean, this is what your donations get you, FaceTime with these people. So Bill Kaelin I've known for like 35 years, and if you remember nothing else, you'll remember that he won the 2019 Nobel Prize in Physiology and Medicine. Bill is also a Howard Hughes investigator. He is the Sidney Farber Professor of Medicine at Harvard Medical School and at Dana-Farber, and has been a great friend of mine for many years. Sigal Kadosh is at the other end of the spectrum. Bill's very old, and Sigal is very young. In fact, she was one of the youngest faculty members to be brought into Harvard Medical School. I think you started at age 28, if I remember. You direct the epigenetics program at the Broad Institute. You are also a brand new Howard Hughes investigator. Congratulations, that's a wonderful accomplishment. And, yeah. And the co-founder of Foghorn Therapeutics. All correct. So let me begin by asking Bill. So you've been here for a long time. We heard about lifers. I'm a lifer, you're a lifer. We vote with our feet. What is it about the environment at Dana-Farber that's kept you here for so long?
Well, I actually came to science pretty late. I came to do my clinical training at the Dana-Farber, but thanks to the great, and now unfortunately late, David Livingston, I pivoted towards science. But I think what's true of most scientists is you want to be at the place where you can make the biggest discoveries and have the biggest impact, and where the people around you are going to make you better. I learned the simple truth many years ago: you grow faster when the people around you are smarter than you are and more talented than you are. So what's kept me here is anytime I've had the opportunity to go elsewhere, I've immediately said, well, who are my colleagues going to be? Or who are the young people who are going to entrust their training to me? And I always come back to the same answer, which is I'm already at the best place to do the science I want to do.
Yeah, I think the people is an important aspect of this. How about the sort of physical infrastructure and the resources that are available?
Absolutely. So, I think great people is necessary, but not sufficient to do the research. You need the infrastructure. You need the equipment, which, as Barry has said, has gotten increasingly sophisticated. You need the sort of on-site technical expertise to really enable you to do the kinds of experiments that you want to do. So you're absolutely correct. It's not just the people, it's also the infrastructure.
That's great, thank you. So Sigal, I've hinted that you were disgustingly young when you became a faculty member. And I mean that with respect. But when you began, tell us about some of the challenges as a young faculty member trying to get, you have your scientific vision. How did you realize that vision?
Yeah, so thanks. So first of all, thanks for having me. I'm really delighted to be here and to see so many familiar faces and friends. And as we've well established, I am young. Thank you. And I started extremely young, for this position. And I have to say, I think actually Dana-Farber probably had faith in me and my hopefully ability to develop a program sooner than I had it in myself. In fact, George here, who's sitting right here, played a role in hiring me. And it was a very pivotal moment in my career that really laid the foundation for all the work that we do now. But starting at 28 with really actually only about 3 years of training. Actually, when I interviewed here, didn't even have my degree yet. I do now. Don't worry about it. I do have it now. As I interviewed, it was extremely early on in these days, and that's challenging for an early investigator to get funding that's required really to advance our research. And so, as we started here, and as I started the lab, some of the most critical funding really came from all of you, from philanthropic efforts that supported our high-risk, high-reward efforts in the lab. And as Barrett mentioned in his introductory comments, the NIH, we get grant money from them, but it's often to support really incremental next steps in these types of studies. And that's insufficient to do what we're all here to do today, which is to try to defy cancer. That's not going to cut it. And the kind of work that I was so excited about doing, coming here, really on the heels of one early but exciting discovery that has now really changed the way we look at how our genes are controlled, how the actual topological architecture of our genomes are controlled, was set to do. And without philanthropic support, we wouldn't be here today. We wouldn't have been able to, actually almost take several steps back before we could run forward with, kind of trying to, push forward a new area of research and a totally new way of thinking about cancer. And so it's critical to have relied on that funding.
Yeah, thanks, Sigal. That's an interesting insight. Bill, maybe we can get a little more granular about costs. What do you think are the biggest sort of misconceptions about research funding and research costs? Really the cost for us doing what we need to do.
Well, I look back at when you and I were getting started and in the lab, as you know, frequently you might study one gene at a time. And frankly, for some biological questions, that was fine to focus on your gene. But for many biological questions, that's hopelessly inadequate. You really want to understand all of your genes, which we have on the order of 20,000 genes. So to use an analogy, if you were trying to understand a symphony, you wouldn't just focus on one musician in the orchestra to understand the symphony. You'd have to have a way of understanding what each of the musicians was doing. So thankfully, we now have technologies to study in parallel all 20,000 genes. We can obtain all the sequences of the 20,000 genes. We can determine in a cell which of the genes is on and which is off at scale. And finally, and equally importantly, for the first time, we have the ability to toggle genes on and off one at a time to see which genes are linked to certain cellular behaviors. So there's been an unimaginable advance in technology, but that technology comes at a cost. And fortunately, with respect to sequencing, the technology improvements have been so robust and frankly remarkable that it cost about the same to sequence 20,000 genes now as it did to sequence one gene when I was getting started. But for some of these other sophisticated technologies, such as monitoring the behavior of all 20,000 genes simultaneously, knowing which are on, which are off, again, toggling them on and off at will, these technologies are expensive. Now, fortunately, they're not 20,000-fold more expensive, but they are 10 to 100x what we used to pay for certain types of experiments. And just to give you a feel for what numbers we might be talking about, routinely now, experiments that might have cost in the hundreds of dollars now cost $10,000, $20,000, maybe $50,000. And they get even more expensive if you want to maybe compare cells to one another or find out how a cell responds to a particular drug. Now, I'm old enough to know that in certain industries, $50,000 is a bit of a rounding error. But I can tell you in an academic lab, $50,000 is real money. It's enough that sometimes, and I can tell you there's nothing more painful to a scientist. You see the right experiment. You know you can do it, but you have to say, you know what? We might have to wait till next year because I'm not sure our budget is going to support this experiment that might cost us $50,000 or $75,000. So I think part of it is the technology. And then the last thing I'll say is, and this is another thing we only dreamed about when you and I were almost as young as Sigal. With this technology I'm sure many of you have heard about called CRISPR, gene editing, we can now make mouse models that are much more faithful models of their human counterparts. So we can actually introduce some of those mutations we were talking about a moment ago rapidly into adult mice and see them get various tumors that we think are a better model for humans. But it's expensive, as you know, to house these mice and take care of them properly. So again, my lab's budget currently, it's about $15,000 a month just to keep the mouse colony going.
Yeah. So Bill talked about all the exciting ways that we spend money to do research, and I just feel it's important for me to point out that it also costs money to keep the lights on. Yes. It costs money for depreciation. It costs money to rent new facilities. It costs money to support HR, and on and on and on. And all of that stuff has to happen as well, otherwise the research can't get done. So let's think about other ways that we get support and other ways that we work with external entities to do the kinds of things we have to do. Maybe you could talk, Sigal, a little bit about the commercial sector.
Yeah, I think this is something that really makes Dana-Farber unique. And over the course of the past decade and increasingly under Laurie's leadership, we really have the opportunity to be exposed routinely to critical players in our collective mission to drive forward new therapies. Now, much of the work that both Bill and I do in our lab is very basic. We uncover the molecular mechanisms governing how cells function. And oftentimes, by understanding how those cells misfunction in the context of disease, particularly in cancer, we can inform how they should function in our normal tissues and vice versa. Oftentimes, some of the most important discoveries that have led to new cancer therapeutics find their center in really true deep biologic mechanism, and that's what we have invested our careers in doing. Now, to translate that into new therapies is extremely expensive and requires a really perfect symphony, if you will, to use Bill's analogy, of individuals across sectors to be able to push this forward. And we're so lucky at the Dana-Farber that we're constantly exposed to members of the venture capital community, members of the biotech community, members of big pharma, members of large corporations, members of philanthropic organizations to collectively come together to advance pushing these drugs or discoveries forward. In our particular case, we made discoveries that arguably were on the most basic of basic scales. How genes are regulated, how genes are turned on and off, what governs which regions of our 2 meters of DNA inside each one of our trillions of cells are nice and open, allowing for genes to be turned off, and which ones are closed. And it turns out that that is a fundamental process that is critical to, by gene mutation alone, in 20% of human cancers, and likely up to 50% of human cancers. So it's something very imperative for us to study. And each time we were in the lab uncovering another rock, so to speak, we realized there was a therapeutic opportunity accompanying that new basic mechanism. And so one of the reasons I came to Dana-Farber is this community is unparalleled in establishing those intellectual adjacencies to put the right people together at the right times. And we now have accelerators, in fact termed accelerator programs, to be able to more quickly recognize those early basic discoveries at their earliest conception and be able to rapidly take them forward toward new strategies for treating patients. And that's something that's deeply inspiring to me every day. And everybody comes together to make that happen. The patients, the staff, my fellow faculty, and everybody else we've been able to get involved. So this is really exciting.
Thanks, Sigal. Bill, you've been at the sort of the VC level looking at this as well. Any other comments about this?
Well, I would just echo everything Sigal just said. I will say, and this is, I think, a change for the better, I'm sure you would agree with me, that, again, back in the days of the dinosaurs, when Barry and I were still doing experiments at the bench, you know, it was white hats and black hats. If you were in academia, you had a white hat. If you were in industry, you have a black hat. But, you know, that was kind of silly. But those barriers are broken down. And as Sigal said, it's a win-win, because there's things they do better than we do, frankly, in terms of the actual translation and getting the ball over the goal line, whereas we do the early fundamental knowledge generation that they really depend on. And the other thing that's happened over time is that the entire biomedical ecosystem in Boston has really, I think, by any account, emerged as the #1 place to do biomedical research in the country. So you can think of it as akin to Silicon Valley for doing computer science. So it almost becomes self-fulfilling that some of the best and the brightest young people who want to do biomedical research come to Boston. And frankly, if they're interested in cancer, they're beating their way to the Dana-Farber. But it's because we do have this ecosystem with lots of really vibrant biotech companies, pharmaceutical companies, as well as these great academic institutions. And the last ingredient, as you kind of alluded to, is, you know, the VCs sense this too. So they want to be here because this is where the action is.
Yeah, the action is here. So I'm going to exploit the fact that we have two of the most insightful and brilliant faculty members up on the stage and ask you in just a minute or two to tell me what's the next big idea? What's the next great thing that you see right over the hill, Sigal?
So I'm going to flip the question just a little bit, if you don't mind. And I'm going to say, what's the current big thing? And one of the most important current big ideas, I think, is leveraging all of the genetics that has poured into our research community over the past now decade. The sequencing, as you all know, of the human genome provided striking advances in understanding the fundamental underpinnings of human disease, and particularly of cancer. And we now know for the first time the essential ground zero of many cancers. And as biologists, that gives us a very important foothold to be able to start understanding those mechanisms, understanding how mechanisms go together. We heard from Judy Garber, there's some of these important cancer-causing genes that she mentions as part of these important clinical genetic studies. We now have hundreds, thousands, that we now have to interrogate. How are we going to do that? You can't just do that by looking at the genes. You have to understand the pathways, the proteins that they're part of, the protein complexes, the conglomerates of cellular entities in which they function. And that's one of the things our lab is fiercely committed to. The more we can understand the actual structure, if you were to look inside the cell, what are the structures of the elements that are actually perturbed, the better we can understand them, and the better we can leverage the tools. As Bill mentioned, it's not just sequencing to understand the genes, it's sequencing technologies that actually enable us to, for the first time now, screen and score the 20,000 genes that are on and off in each one of our cells and determine what are the dependencies, what are the vulnerabilities in certain cancers that are specific for those cancers that we can go after. That's enabled by sequencing. CRISPR is enabled by sequencing. A number of technologies have been enabled by this. So I would say the next big idea is actually addressing the flood of new data that we have and being able to put a meaning so that we can stratify patients and best understand how we can advance personalized medicine.
Bill?
Well, I always preface my answer to this question by saying I'll be really disappointed if I'm right, because as a scientist, I live for that unexpected result, that secret of nature that just completely shatters the way I thought about medicine, thought about biology. And so I hope I'm wrong on this and that there's a great discovery around the corner that none of us can anticipate. But since you forced me to give an answer, those 20,000 genes only occupy about 2 to 3% of your total DNA, or what some people refer to as your genome. And so for years, we've referred to the rest of the stuff as sort of the dark matter of the genome. But you can be sure that most of the variation you see in this room is due to subtle changes in the dark matter. So we have to understand the dark matter much better. And I'm sure the dark matter is also going to play a very important role in cancer. In fact, I think there's already pretty clear evidence that it does.
You know, we have another minute or so, and your description of how you hoped you were wrong just reminded me of what David Livingstone used to say when he'd see some great thing. You know, he'd say, "Buddy, it blows my mind." So maybe talk a little bit about David's influence on you. About how to run your lab, because that's what we're talking about today.
Well, this is hard for me because many of you know I love David and owe my scientific career to David. But David was all about, you know, curiosity, having scientists follow their instincts, follow their noses, and then he was absolutely passionate and uncompromising when it came to arriving at the truth.
Yep.
And so of the many, many things David taught me, I think that's what's most important to me, is having that kind of a role model, to see his dedication and commitment to the truth. And I think, like most physician scientists, in the end, he was motivated by our patients. And all you have to do is walk through the waiting area to know that what you do really matters. And so, David was just, I couldn't have asked for a better role model.
Yeah. Thank you for reminding us all about David. So I think we can move to the question period now. And just like before, people will be walking around with microphones. So I can't see very well, but if there are any questions for the panel, take advantage of this,
I would ask you, Bill, I love this story. You know I love this story, but how you went from college to becoming a Nobel laureate.
Okay, so
really interesting story.
So the Reader's Digest version is that when I was a pre-medical student at Duke, I was doing pretty well on my grades, but someone whispered in my ear that if I wanted to get into medical school, it would look really good if I worked in a laboratory. So I met with a chemistry professor who told me he had a particular project that 7 undergraduates had worked on before me, and all had gotten into medical school. So of course, I was very happy I had found the golden ticket and immediately signed up to work in this lab. Now, if I was a little bit older and wiser, I might have asked, well, why couldn't these 7 people bring this to completion? So naturally, I became the 8th person who couldn't bring it to completion, and then I had the audacity during my last lab meeting to say, I think this project will never be brought to completion because I think it was based on a mistake by one of my predecessors. And as professors often do, he rewarded me with a C-, which for a pre-med is like having a wooden stake driven through your heart. And then he hand wrote in the margin of my Duke transcript, "Mr. Kaelin appears to be a bright young man whose future lies outside the laboratory." So, which is like being told you have a good face for radio. So that was actually the beginning of my scientific career. But the C- did succeed, and so I did get rejected from Harvard Medical School, and I was rejected from Harvard for residency. But one day, Bob Mayer called me. I don't know where Bob's sitting. Bob called me to say that I had been accepted for the oncology fellowship program. And at this point, actually not going to Harvard, it always sort of turned out pretty well for me. So I didn't know whether I was making a mistake or the Dana-Farber was making a mistake. But I came to the Dana-Farber to be a clinician, partly because of that terrible experience. But after a year and being surrounded by all these great minds and all these great scientists, and just by dumb luck winding up in David Livingston's lab, my life was changed. So that's sort of the nonlinear path to Stockholm.
Question over here. I think you can see me. We often hear about labor shortages right now in various parts of the economy and various parts of the hospital even. And I was wondering if you could comment on the supply of talent in your field. What are the challenges? Are you finding enough? And what we can do to support that effort?
Yeah, thanks. I think it is an issue at the moment, and I think actually part of this stems back to what Bill mentioned earlier, that it used to be that there were black hats and white hats, right? And there were some, you would either go into academia and you would stay in academia, or you would go to industry. And now, which is a very positive thing for the field, there are actually numerous career paths that our students and trainees can take. And I think that's important because, as Bill mentioned, we can't do it all ourselves. In fact, if our role here is to understand the basic mechanisms, you don't want Bill and I trying to push forward now the clinical development and all the work that needs to identify a lead compound and ultimately go into therapeutic territory. So I think that we have more fields that these talented individuals across the world really can go into. to. And so for that, we see more of a distribution, I would say, of people coming from outside the country, within the country, even just having various different career paths, because they actually exist now. There are a number of different career paths. Biotech, I have a very wide, diverse mixture in my own academic lab of 25 people or so. Some who've come in actively telling me that they can't wait to be the next venture capital investor in biotech around Boston, and others that fiercely are going to do everything they can to have my job and to be able to run a lab. And I think that's part of what we may, as investigators, perceive as a shortage. But it could also, for the industry, also mean increased interest in just a much more wide repertoire of possible outcomes for this exciting time in science. Yeah.
Have you been seeing visa challenges?
Well, I think even before the visa application, I think unfortunately there is a perception that the United States is not as welcoming as it was during the 60s, 70s, and 80s. So there's been a fall off in the number of applicants from abroad. As Sigal just mentioned, also this is the dark side, not dark side, but the alternative view of the growing biotech industry, that they do now hire people very early on, even in many cases without even doing a postdoctoral fellowship. And then the last point is federal funding has been flat for so long that I think it's finally caused some people to redo the calculus, because you may know that these wonderful postdoctoral fellows who we rely on so heavily are grossly underpaid, but historically they've been willing to do that if they could turn around and become an independent tenure track investigator and have a reasonable chance of getting a federal grant to start their laboratory. But they're seeing now that maybe that assumption isn't always correct. And so we're having much more attrition than I think we ever had before.
To end on a positive note, I'll just say that one beauty of being at Dana-Farber is just the diversity we have among our investigators. And I think talented people will always flock to a diverse academic institution. And because the mechanisms governing cancer are so fundamental to so many different biologic processes, we fortunately are able to recruit scientists that have training in an incredibly diverse repertoire of biologic and other scientific disciplines, from physics to chemistry to life sciences. That won't cease to be the case. Cancer requires all those disciplines to be able to traverse an incredibly challenging series of questions that we have to tackle to be able to answer and to defy cancer, as what is shown here on this slide. I think that we continually have to make sure we're attracting that kind of talent across the different disciplines. And even within the scientific disciplines, we house at Dana-Farber likely the world expert in each of these different areas. And that's something that I don't think another place can say, and that's something that makes it incredibly rewarding, stimulating, and inspiring to work here.
So that's incredibly uplifting. I'm going to bring the room down again.
I tried.
Because one of the great things that's evolved is that in addition to the academic individuals that we hire, postdocs, graduate students, and so on, Dana-Farber has evolved a system of hiring technological experts, people who do experiments because they want to do the experiments. There's an analogy, unfortunately, to these kinds of positions in industry. And what we've seen is that we cannot match current salaries in industry. And so we train these people up, we give them experience, and the next thing you know, they've left for 30, 40% higher salaries. But there is a group of people who stays, and that group of people are the ones who buy into the mission. And there are people who are willing to take the sort of mission-related discount in their salaries to be able to stay at Dana-Farber. And you would think that it was just the physicians or the scientists, the academic scientists, but no, everybody finds a way to sort of buy into the mission and are willing to do what it takes to be able to stay there.
Yeah, I think Deb has a question. Oh.
Could someone get Lori a mic?
I would also add, though, that, and you'll see this in the next panel, our hematologic oncology fellowship is far and away the best in the world. And there are such talent among those 14 to 16 fellows each year. And the majority of them want to stay at Dana-Farber because they think it's the best cancer center in the country. And we can't afford to have all of them. We just don't have enough funding and enough space to keep. We only can keep 2 or 3 of them every year. Now, it's not so bad because they go elsewhere. They go to Memorial Sloan Kettering, or they go to MD Anderson, or whatever. But obviously, we'd like to keep more of them around. So we don't have any deficiencies in the number of really talented young people who want to do research at Dana-Farber.
I think Deb in the back has a word. Bob.
Bob.
Could you explain what the value and why it's so important to have a relationship with the Broad and the Bridge Project and how they started?
Bill?
So getting back to powerful technologies, the Broad Institute did invest in certain technologies that are complementary to the technologies we have in-house. And so rather than being duplicative, there was synergy working closely with them. We make them better, they make us better. And likewise with the Bridge Project at MIT, engineers and scientists just think of problems differently. I remember the first time I met with the engineers at MIT, it turns out that they were hammers looking for nails, and we were nails looking for hammers, so it was a beautiful thing to have us in the same room, and then we were able to start solving some problems.
To add to that, I co-direct the epigenomics program at the Broad, in addition to my role here at the Dana-Farber. And as Bill mentioned, it's really incredible synergy, I think, between the two groups. But I will say, if you think about the body of faculty that speak in our seminar series and all of our have our projects, a large number of them certainly are, again, centered here. And I think that comes from my earlier comment about just the diversity of scientific disciplines that must be brought in to study cancer. And that oftentimes, has propelled forward actually findings and discoveries that have changed the course of disease for totally other, different diseases because of findings that we have in cancer. So I think, our collective mission here at Dana-Farber is certainly to defy cancer. But let it be known that many of the mechanisms that are fundamental to what we study in the lab are very likely to also, along the way, impact in a very meaningful way other diseases as well. And I think that's just an example of where we've been able to have the fortunate partnership with not only the Broad but other institutions around this area.
Thanks, Kaelin. With that, let's thank the panelists for their insights.